So welcome everyone for this session 2 of Science and Dialogue. We will start the session at 1 and waiting for people to join. We are expecting about 30 people today. And Emeline will start with an introduction before we start the discussion.
I can maybe start sharing the first slide. You okay?
Yeah, of course.
I let you give me the green light, Lila, when you want we start.
Yeah, just maybe 1 minute for people to join and then, yeah.
Sure. You let me know.
I think we're good if you want to start.
Yeah. Okay. Thank you very much. So hello everyone. I am delighted to welcome you to the second session of the panel discussion series Science and Dialogue that aims to foster interactions and discussion around non-animal new approach methodologies or NAMs, regulation and toxicology. Before introducing the panelists of the session and the topic of the day, we wanted first to quickly present our scientific committee ProAnima. Created in 1989 under the honorary presidency of Professor Théodore Monod, ProAnima Scientific Committee aims to contribute to better scientific research and evaluation of medicines and chemical substances for health and the environment from a scientific and regulatory perspective, thanks to innovative methods alternative to animals. To this end, ProAnima has multiple level actions: raising awareness with workshops, public affair work, financing through the DVES prize, for which the applications are open for the 2025 edition, publishing, the unique French review dedicated to non-animal methods, and fostering dialogue, bringing together researchers from academia, industry, and members of regulatory bodies and government, as well as partner institutions and NGOs, such as through our NAMS network and this panel discussion series. So 2 months after our first panel discussion on the regulatory acceptance of NAMS and the approach to define adaptive criteria, we wanted to explore and discuss current challenges, opportunities, and ongoing actions in favor of harmonizing NAMS to ensure it can and will be regulated, accepted worldwide, and under the same criteria. While we see a global trend in international legislations and bodies toward the acceptance of non-animal methods, there is a clear area in terms of testing guidelines that are still very slow to validate the new methods and let down or remove the no longer relevant traditional testing methods. Regulatory agencies, scientists, and all the stakeholders clearly wish for an harmonization of both guidelines and acceptance criteria. We can then wonder how this can actually be possible and implemented among countries, sectors, and disciplines. We thus wanted to ask the following question: harmonization of norms, can it really be global? I won't be any longer before introducing our panelists and moderators, just always address our warm thanks to our partners from the European Project Parc and its French hub, Pascal Sender, Sébastien Denis, and Margot Desplanches, the FRIAR French Center FC FRIAR and its director Athanasios Sotiropoulos, and the ATC and Professor Jean-François Narbonne and Julien Narbonne that support our initiative. So today we are very lucky to welcome 3 panelists that will be able to share their valuable and unique experience from 3 different regions. We have indeed the great pleasure to welcome Dr. Nicole Kleinstreyer, Director of the NICEATM, the US Federal Resource for Alternatives to Animal Testing, and the Executive Director of the congressionally mandated ICVAM. Her work is centered on domestic and international efforts to develop novel testing, modeling, and analysis strategies that provide more rapid, mechanistic, and human-relevant prediction of potential environmental chemical hazards. A warm welcome to you, Nicole. We are also very much delighted to welcome Dr. Surat Parvatam. Surat did her PhD in cell biology, where she studied how cell shape and tissue patterns can influence development and disease using biophysical tools. Post her PhD, she worked as science writer for a few years and published science articles in places such as Nature India, skidev.net, and more. Until 2023, she was the chief manager of the Center for Predictive Human Models Systems, Atal Incubation Center, Center for Cellular and Molecular Biology, the first science and policy center in India that works toward enabling human-relevant research in India. Currently, she works as senior strategist at the Human Society International India. A warm welcome to you, Surat. And the same feeling goes to Dr. Nina Hobby, who is the founder, co-CEO, and scientific director of Alveolix, spearheading groundbreaking advancements in organ-on-chip technologies in Switzerland and beyond. Nina is pioneering advancements in organ-on-chip and NAM technology validations, with multiple publications and honor in the field. She secured a $20 million in dilutable and non-dilutable funding, fueling transformative research and development initiatives, and orchestrated the creation and cultivation of a dynamic research and business team, fostering collaboration, innovation, and a shared dedication to excellence. Thank you very much for being here, Nina. The session will be co-moderated also by 2 dedicated experts, Professor Merel Rietke-Wattinga, Professor of Evidence-Based Transition to Animal-Free Innovations at Utrecht University's Faculty of Veterinary Medicine. After a graduation from Utrecht University as a veterinarian, her work has been dedicated to the FRIARs in first instance, and since 2008, she has been promoting the methodologies of systematic reviews for animal studies. In 2012, she founded CIRCLE, the Systematic Review Center for Laboratory Animal Experimentation, and she and her team organized the first international symposium in the field of preclinical systematic reviews. The evidence resulting from systematic reviews of animal studies have been confrontational, which made her decide it was high time to make the full shift toward replacement and the evidence-based acceleration of animal-free methodologies. Thank you very much, Merel. And Dr. Lila Courteau, scientific head at ProAnima. Soon after obtaining her PhD at the Cancer Research Center of Toulouse, Lila decided to focus on promoting good science through developing an expertise in non-animal methods. As our scientific head, Lila has launched and is moderating our international networks of experts in NAMS, this panel discussion series, among other things. She is also representing ProAnima in the Global Education Hub, recently launched by the TPI and PETA. Thank you very much, Lila. And before we start, just reminding you that the session is recorded. We will now have a 35 to 40 minutes of discussion. We will try to have a good 15 minutes for Q&A at the end. You are invited to ask and write your questions related to the topic during the session in the question section of the platform at the bottom right of your screen, with the idea and possibility for all of you to upvote the questions you really want to see addressed by our panelists during the Q&A. For any technical issues or to share links or reference, you can use the chat sections just next to the question section. I think we are now good. Thank you all for your trust and for being here with us today from France, across Europe, but also from Asia and America. We are very happy to have you. And I now give the floor to Lila and Merel and our panelists for a great session of discussion. Thank you very much.
Thank you so much, Emmeline, for the nice and clear introduction. I am indeed very much excited and honored to be able to moderate this discussion. And as a first question, and to help setting the scene, I wanted to kindly ask you, the three of you, to provide a quick overview of the current state of regulation and use of NAMs in your country. So if one of you wants to start, please go ahead and then we can proceed. Otherwise, I can pick randomly. So up to you.
I am happy to start because I think I'm on the left side on anybody's screen. Quickly, thanks to all of you to organizing this. I feel I'm really looking forward to this discussion. And I think the first question is already picking something where it's very challenging, because I think this is one of the most limitation. I'm sure Nicole has much more information about this, but it's one of the most limitation to know what actually your country is already involved and who is the real responsible person for NAM, and how much is already kind of driven to the regulatory bodies. Because in Switzerland we have Swissmedics. This would be our regulatory body, but on the other side you also know everybody who is in biotech. Maybe you're not doing your first clinical study in Switzerland because it has very high portfolios, so maybe you go in other country. So Switzerland is maybe not the one who is the most advanced when it comes to NAMs. Netherlands is way more further, and there are, beside that, Netherlands has a lot of knowledge and a lot of driven for NAMs. Also, Netherlands has a very straightforward clinical trials at the early stage, so they also have a lot of possibilities for clinical trials. So a lot of stuff is going on in Netherlands, and this is really what I see in Switzerland. We have now a nice, because we have the 3R Center Switzerland, who is really driving this. We just had a nice yearly event with them. We have Swissmedics, who are very open to discuss, but they are waiting for more order, maybe from bigger regulatory bodies from other countries. And we have on the other side, we as developer who would like to interact, and this is great. The door is open, the government is listening, Swissmedics is listening, but what is still missing is what we're doing now, what Switzerland is exactly doing now. And I want now to give the word forward, because I know there are other questions coming in this direction, and yeah.
I'm happy to pick up the ball and keep it rolling. Okay. So in the US, we have a number of drivers for implementation and adoption of NAMs. They range from existing standing committees to legislative mandates. So the committee, ICVAM, the Interagency Coordinating Committee for the Validation of Alternative Methods, is comprised of 18 different agencies. We actually keep growing steadily. So most recently, the National Center for Advancing Translational Sciences requested to become a formal member of ICVAM. And so ICVAM has within the committee not only research agencies, but regulatory partners. So obviously the two biggest regulatory agencies in the US are the US Food and Drug Administration, the FDA, and the US Environmental Protection Agency, the EPA. Both of those agencies have really stated their commitment towards transitioning toward use of NAMs wherever scientifically justified and wherever feasible. The EPA in particular has, historically put out some timelines and some very, very clear objectives for replacement around, for example, the acute toxicity 6-pack. So the 3 acute topical toxicity tests of skin and eye irritation and corrosion and skin sensitization, and then the 3 acute systemic toxicity tests by various routes of administration. And I think, and then of course the FDA is driven not only by their own internal commitment, but by recent legislation such as the FDA Modernization Act 2.0, which really opens the door for submission of NAM data in support of, for example, new drug applications. And so I think one thing that has supported the success of regulatory implementation of NAMs in the US is that dedicated organization, and not just the committee of ICFAM, but NICEDM. So NICEDM is the center, the national center that is part of the National Institutes of Health that provides the operational and scientific support to ICFAM. So ICFAM is a committee that does not have a budget. All of the agencies participate in ICFAM in an in-kind way, and they provide really substantial contributions in terms of time and energy and resources. They're subject matter experts participating in monthly teleconferences and on work groups. But it's absolutely critical to have this other national center, NICEDM, which has a budget of around $6 million a year, to support the priorities and the objectives of ICFAM and projects that really have a high likelihood of advancing the science and the regulatory implementation of NAMs. And that's really how we prioritize what NICEDM works on. It's what's highest priority for our ICFAM agencies. Where is the science really ready, and where can we make the most impact? So the websites that we maintain around NICEDM and ICFAM have really a wealth of information around the current state of regulation and use of NAMs in the US.
I can go next regarding India. So in India, the Minister of Health and Family Welfare passed a landmark amendment last year, where they amended the new drugs and clinical trial rules, which now allows the use of non-animal methods for the drug testing and safety assessment. So it was a very landmark move made by the government. And India has already banned the animal testing in cosmetics, and also the import of cosmetics that have been tested on animals in 2014. And other than that, in the agrochemical sector, so in 2017, they had released a set of guidelines. It's not a law, but it's a set of guidelines where the document does recognize the use of any OECD approved alternatives for testing of agrochemicals if it is approved by the scientific committee that looks at the data. So there have been several advancements in the legislation and regulations that have come in India. In terms of usage, I would say when I joined the Center for Predictive Human Model Systems 2019, there were barely any labs and companies in India that were working on non-animal methods. But in the last 5 or 6 years, I would say that there has been an exponential increase in both the smaller companies, startups, and academic labs that are working on non-animal methods. And there is also a database that the center has created where almost 80 plus academic labs and companies are registered, which are currently working on non-animal methods. So it's definitely a growing field in India, I would say.
Thank you so much. That was super useful, I think, as a good start for then moving on to the next question. So Marianne, if you want to go for this one.
Yes, thank you, Lila. And thank you to the panelists for a great start already. So my question is on what you think are the current challenges to step up on the harmonization of NAMs? And then specifically, how can we make better use of scientific evidence for the NAMs, a lot of evidence is requested at the moment before they become accepted, whereas for animal studies, we never asked for that evidence and it is largely not there. So what are your views on this? So feel free to take the floor.
I would love Nicole to start there, because as you guys are really most advanced, you know, I can then also give a little bit the inputs what is the challenge on other countries where we are not already have such organization and such also funding for doing harmonization or for accepting something. So I'm happy that you start.
Sure. Yeah, absolutely. Thank you. So I think there's a number of factors that are challenging. And like with most areas, it really comes down to resources in one shape or another. So resources for validation of NAMs, resources for access to the NAMs themselves. So reliable laboratories that operate under GLP standards that offer standard SOPs and reasonable access to NAMs in different regions of the world. Awareness and communication, so resources needed to support training that will facilitate implementation of NAMs, but also acceptance of NAMs on the regulatory side, so education around NAMs that are available that are actually providing better public health protection, and we therefore have a responsibility to use. Yeah. And simply access to information and just knowledge that these NAMs are out there, that they have been standardized, that they have been validated. So there's quite a range of efforts needed to try and put more resources into each of those spaces. One of the things that we're doing, so I mentioned, we have a website, the NICEDM website has a page on it with about 100 or so methods that are already accepted by US agencies, but that's just a static list. I mean, we keep it very up to date. Our communications manager is amazing. However, we are actually launching a new initiative in the US that was requested by our federal agency partners to develop a new interactive resource and database of alternative methods for regulatory application. So that would be not only providing information on what methods are available for what endpoints and what agencies would accept them for what different types of substances or chemistries, but also allowing users to access wherever possible the scientific evidence. So the data that was used to validate those methods, be able to apply things like cheminformatics workflows to look for similarities of a user's substances to the substances that were used to validate the methods, the applicability domain of various methods, to make data-driven selections of what methods may be most appropriate for their use cases and for the regulatory agencies that they want to submit to. And there are other such applications. For example, the JRC maintains an application called TSAR, so the Tracking the Status of Alternative Methods for Regulatory Use, which is an outstanding resource as well. And we coordinate with our partners at ECVAM and elsewhere around the world through ICADM, the International Cooperation on Alternative Test Methods. Mm-hmm. And so creating parallel resources in the US is something that we're focusing our attention and our resources on right now. And then I think there's also a real need on the part of funding bodies to demonstrate commitment towards the importance of validation and qualification. So not just fun, exciting, cool research, but actually taking these new methods and taking them through the pipeline of assessing their transferability and technically characterizing them and ensuring that they're rigorous and reproducible in a way that will support their acceptance by regulatory authorities worldwide. And there's a new program in the US that's just been launched by the National Institutes of Health, the Common Fund, which is a large sort of high risk, high reward grant funding program out of the NIH Office of the Director. And it's called Complementary, so Complement Animal Research and Experimentation. And it is focused, it'll be several $100 million over at least 5 up to 10 years. And it's focused on the development of human-based NAMs in biomedical research, but there's a very specific aim that's been outlined under this program for validation and qualification with an eye towards eventual regulatory implementation. So I'm delighted to see that commitment on the part of NIH, which is, you know, the largest funder of biomedical research in the world. Obviously, there's parallel efforts in the US and the EU under PARC and other such initiatives, but I'll let others speak to those.
Yeah, Nicole, I have one additional question, because in the previous answer you mentioned that NICATM gets $6 million per year in funding, which is fantastic. Also, the work you're doing, is that government money? So yes. Should that be an advice to everyone to have institutes like yours?
Yes, that funding is part of the budget of the Division of Translational Toxicology, which is part of the National Toxicology Program, and within the National Institute of Environmental Health Sciences, that's part of the larger NIH system. So it is government funding.
Okay, wonderful, thank you. And Nina, you want to continue?
Yes, very happy. I think Nicole ticked nice spots. And also your question, I wanted to ask before exactly this. I think this is currently, and I now not just talk for Switzerland, also the ecosystems that I know very well is Austria and Germany. I think this is missing, that we have actually an independent funded institution for NAMs. So we really, I think this is, and also I think there we come back to the first question, we can do global. Currently, I think America, US, North America is just funding way more, really like in the implementation, than Europe. So as for everything, we a little bit trying then just copy paste, which is great because on one side it's cheaper for you, but on the other side, maybe it harms a little bit the harmonization over the whole global aspect, because we are not working in the same pace, I would say. So this is really something which I say EUs definitely have to catch something up. There are nice programs now putting up from the EMA. It's about the ITF program, for example. It goes in this direction, but exactly the most important part, money, is missing, and therefore the pace is not that quick. So nationally, as you said, Muriel, it would be very good. We have, for example, an SNF grant, which was about NAM implementation. Yes. But SNF is really responsible for academic research. So what you start, what also Nicole mentioned, is that again, it goes a little bit in this high impact research versus giving the money to the people who really starting to make submissions and filling the databases. So this is missing in Switzerland currently. So the awareness is there, and I think it always comes step by step. First, it goes in the basic research or in the academia, and now they realize, it's too less implemented. So I hope InnoSwiss, for example, it's a very nice funding body in Switzerland, which really goes to the companies, but you have to work with academia. So this could be a stage that then regulatory submissions are really done, because biotech would get funding that they can do this extra or replacement of non-needed animal testing. This is a point which I really see where we have to pick up, and where you say it has to be governmental money, because then it also gives an impact. The second part where I think also want to go for harmonization. Nicole said about databases, and I really see there, we as a company, as a small company, we have really a lot organs on chip data already in IND studies, because we have partners who are really forward thinking. So we have biotech partners forward thinking, plus they had the limitation that their compound was not able to be tested in animals. So they anyway were depressive. They needed to have a solution. Of course, it's making the case much easier. So FDA approved them that they will do a strategy without animal testing. Makes the case much easier, because they're not losing this opportunity. Or maybe if we go with the animals, we are safe because the regulatories will accept this, versus with the NAM, we have to explain what we did. So this gives us a little bit, I think there is a nice corner where we can make data. And what is then missing, that when someone is submitting that exactly such database like Nicole's team is filling, that the biotech company can click and say, I used NAMs, because they are the one who knows best what they submit. So maybe the regulatory bodies, they would even not understand that this was a non-animal approach, because they just make the revision and don't understand, this part was non-animal approach. So therefore, I think there is this would be tools to make harmonization, because we really know for this molecule, this was done. And if everybody would click this by themselves, they know best which molecule they were using.
Yeah. And that would also require cooperation between scientists and regulators, right? So we get the proper stuff in the databases. Yeah. Thanks a lot, Nina. And then Surat, we lost you a moment. So you're back, fortunately. So do you want to comment?
Yeah, sorry. I missed the question, so if you could just repeat that.
Yes. What are the current challenges to step up for the harmonization of NAMs, and how can we make better use of evidence, scientific evidence?
Right. Yeah, definitely. Like you mentioned, one of the biggest challenges is that all of these different stakeholders, regulators, technology developers, they're all currently working in silos. So there is definitely a need to kind of increase the crosstalk and engagement between the different regulatory bodies and the government bodies, the funding agencies and the technology developers, because we also spoke to several, for example, private investors who have no clue about what non-animal methodologies are, right? So if we need to look for increasing private investment, so we also need to increase the awareness amongst all these different stakeholders, regulators, funders, and we need to devise some kind of engagement mechanism where we can bring all of them together and have regular modes of crosstalk. So another aspect is, for example, even like the regulators are not really aware of how to assess or how to understand these technologies. And I mean, this is not just a situation in India. Like, for example, we had a meeting 1 or 2 years back where the regulators, it's just a question with both the technology developers and the regulators on what is the best way to, what is the acceptance criteria of these technologies. If someone does submit some data to us, how are we supposed to assess it? So I think these are all the challenges which, again, are not limited to India. So we need to come together globally with all the international regulatory agencies together and agree upon a set of acceptance criteria and like what are the minimum acceptance criteria. And again, because MPS are so fit for purpose, right, we can't have a generalized set of criteria for all the methods. So we would probably need to come up with prioritized 1 or 2 endpoints. Again, coming up with prioritized endpoints at a global scale, again, could be difficult because some countries might have their own priorities. For example, some diseases or some endpoints may be more important for India, where the unmet need is higher compared to some of the other countries. So again, the first step would be to maybe identify 2 or 3 priority areas where we could come together, where all the regulatory bodies or some international agencies could bring these regulators together and release a set of guidelines for at least 2 or 3 endpoints. And having these success stories is very important to build the confidence in these methods. I think that is one of the biggest hurdles, which I feel even more than any other aspect, is because even if you cross the regulatory hurdle, you need to build the confidence in these methods amongst the regulators, technology developers, even private funders, because if they are going to invest their money, they want to see the return of investment. They want to see the product successfully pass the stages, so then only they're going to invest. So building confidence in these methods would be one of the biggest hurdles which we would have to cross, and that we'll probably have to work together, bring out certain success stories, bring out guidelines, and acceptance criteria, which is mutually agreed upon. They are not just guidelines which have been published and everybody forgets about it. So it should actually be everybody should agree upon those set of guidelines and methods. So yeah.
Okay. Thanks a lot, Leela. Before we move on, I was just thinking about validation and qualification. I learned that from the Noura recording from Nicole, that validation and qualification are actually kind of the same terms, but the FDA has moved away from the wording validation, if I understood correctly, because it was a tick box exercise, and so they prefer the word qualification. And I thought that was very enlightening because I had that question for a long time. So then if that's correct, what I've understood, Nicole, then we move back to Leela.
It is correct, but perhaps if I could just expand on that just a little bit. There's guidance, there's quite a lot, and this is probably actually a nice segue into the next question about sort of accelerating the process and international work. But in the US, we've recently published earlier this year the ICVAM report on validation, qualification, and regulatory acceptance of new approach methodologies, which is really intended to be a guidance document on how to consider and implement some key concepts in a scientific confidence framework that will support regulatory implementation of scientifically robust and high confidence strategies. And one of the concepts that we try to convey in that report is that there is no such thing as a one-size-fits-all approach to validation, that you have to consider the context of use, as Surat was referring to, and ideally that method developers and people who are trying to validate methods will have communication with the intended end users, whether those are in regulatory agencies or in regulated industry. They will have that communication early and often throughout the course of that process to ensure that feedback from those stakeholders is being taken up as they're designing validation studies, as they're going through qualification exercises. And we have a new initiative. It was just announced, I think yesterday or the day before, in our NICEDM newsletter, called the Method Developers Forum, which is intended to provide exactly that opportunity for communication between method developers and end users. And we are this is going to be the first of several in a series, kind of lasting indefinitely, where we focus on particular endpoints. And so the first endpoint is carcinogenicity. So we're asking method developers that have methods that are relevant for screening compounds for carcinogenic effects or contributing to hazard or risk assessment for carcinogenic endpoints to submit their proposals to us to participate in this Method Developers Forum. We're asking them to consider the key concepts in the ICVAM validation report as they're structuring their presentations. But also, and I think this is really important, we have worked with our agency partners and our industry partners to pre-record needs presentations. So we have presentations from various offices in the EPA, from various centers in the FDA, from other agencies that explain how do they currently go about assessing carcinogenic potential of the substances or the chemicals, chemistries that are within their regulatory jurisdiction. What kind of data do they use? How do they consider that data? How do they join that data together? Where are there gaps or opportunities for improvement? And having I think a real understanding of the current process and the goals of that process in terms of public health protection is fundamental towards creating a foundation where we can then build methods and models that are intended to be fit for those purposes. And so, like I said, the presentations that are pre-recorded that are going to be available on the NICEDM website are just the first in an iterative series. These are dedicated toward carcinogenicity, but in a few months, we'll have another event focused on developmental toxicity or reproductive toxicity or cardiotoxicity. Sort of the list is endless, but we're trying to really facilitate that interaction.
Yeah, that's great to hear. So back to Leila.
Thanks. Yeah, thanks for all this detail, Nicole. I think it's really useful to know what is actually happening and implemented. And thank you all for raising all these very good points on the needs for resources, budgets, and communication. I think we all agree that it's basically what we need. Meryl, I was thinking that maybe we can kind of mix the 3 and 4 questions because so we touch base a bit on the acceleration of the process, and I think all the points you raised are part of this, need to accelerate the process. So mainly, I just had one slight question on the legislation. So you mentioned, Surat and Nicole, that in the US and in India, you can now use NAMs instead of animals for drug development. It's not the case in Europe yet. So my question is, do you think that this difference in the legislation is one of the biggest hurdles for the harmonization, or maybe not? Maybe it's not the thing that is the most necessary.
Maybe I can comment on that quickly. For Europe, maybe it's not written, but everybody does it. When we are in Europe, our first INDs were going into Europe regulatory bodies. It was not directly going to FDA. So I think what Europe maybe is missing a little bit to make a nice modernization statement about this. But it's really like more, this is also what we have to face now a little bit, that actually, and I think this needs the process and the acceleration. On one side, we want to have harmonization because this, I would say, is the holy grail. But on the other side, it should not keeping us for feeding the system, because we have to compete with more than 60 or 100 years of animal studies, which maybe the reason why animal studies is still so present is because we did harmonization there, so people don't want to let go from all the effort was invested there to have clear guidelines. So we have to compete with this because people the human being likes to have guidelines. It's nice to know what you have to do, that you get a certain aim. But on the other side, I think we also should not wait for it, and we should produce data. And I think we get now all open doors. Nobody tells us no. Regulatory bodies are open. Government. The only thing what definitely helps is funding. And there, I guess, maybe Surat, it's not so easy in India already that you have this public awareness. Switzerland now changed a lot also with all the vegan. So there is a lot of animal welfare now going on in Europe. So this, of course, helps, because then also the government is pushed to make a commitment, because our tax money goes in something which we don't want anymore. As soon as this starts, then of course it gets easier that tax money gets free also to change this. So this is on the European part. I just don't want that people think in Europe people are not allowed. No, we are allowed. We just have to do it, and we need a biotech partner who's ready to take the risk. At the moment, you cannot test in animal. You don't take a risk, because the only feedback that you get is you have to find a good replacement. So there's not a lot of risk. But if you have a nice guideline for what you have to do in animals, 5 mice, double gender, and you have no guidelines, you maybe go with the nice guidelines. And this is currently what we have with young partners, with smaller biotech. They want to go in on animal, but it's not so crystal clear, so they also take a risk in doing it.
Then should Europe take the lead? And I just remember a good example from Europe. That's the monocyte activation test, the alternative that can replace the rabbit pyrogen test. And it's now incorporated in the European Pharmacopoeia. So that's a great step Europe has taken. So and then Europe is preparing a roadmap on phasing out animal studies for chemical safety testing. So we don't know what's exactly going to be the content of that. But can Europe take the lead, or should it follow the global movement, because we're a global community? What is your view on that?
General or me?
Yeah, continuing with you first, please, Nina. Yeah.
I think it's just like, I think, but maybe I'm too naive in that, but I really think that Europe currently just missed the moment that they make a clear statement. So I think Europe is in many stuff, they are very advanced, for example, for cosmetic or so. I think maybe Europe even started, I'm pretty sure. So therefore, I think it's just about that maybe now it's time that we are not thinking, anybody of us already knows that we go in this direction, that maybe there would be a very clear statement from the EU, from the health regulatory bodies, instead of having single little statements, having a clear statement. I think the Modernization Act 2.0 was so important because it was so exposed, not because it was total new, but people started to read about it and started to realize, yes, we can do. So, but as I said, giving the word to the others, I'm absolutely sure they maybe have also other opinions, because it's not total my expertise, it's my feelings.
But maybe, Nicole, can I ask you, should the FDA or the US take the lead here? Because I think the FDA is determining what the rest of the world is doing, very simply stated. So could they take the lead here to the global change?
Well, I think it's a complicated question that doesn't have a single answer, because I think it's actually dependent on, again, the area of interest and the endpoint. Like, so for particular endpoints, the EPA has really driven a lot of the change by implementing science policy ahead of global test guidelines. For me, actually, if I had to pick one organization that is best positioned to lead, it would be the OECD. So the OECD is absolutely mandated with serving as that centralized resource for international harmonization of test guidelines. And I give a tremendous amount of credit to the OECD Secretariat, to Anne Gormelon, Patience Brown, who have been very, very forward thinking in those, and particularly in the last several years, in terms of approaches to modernize and speed up the OECD process, not just under the WNT and the test guidelines program, but also under, for example, the Working Party on Hazard Assessment, which I think has really transformed over the last several years in terms of the IATA case studies program. There's a new IATA framework template that strives to support sort of chemical agnostic IATAs that can be more generalized and can kind of take a modular approach towards different information sources and can be tailored for specific regulatory contexts, and then can also serve as a breeding ground for defined approaches that would then go into the test guidelines program. There's also efforts, there's a current project on the OECD work plan that's being co-led by the US, the EU, and the Netherlands to revise and modernize OECD GD34. So that's the guidance document on validation. And a lot of the concepts that are being integrated into the revised GD34 are certainly consistent with the concepts from the ICVAM validation report. So I think that OECD is really well positioned to accelerate the process and lead this change. And then, of course, that has to be backed up and supported by individual regulatory authorities and individual regions. Another type of organization that I want to highlight that I think has potential for tremendous impact are consortia organizations. So the newly formed ICCS, International Collaboration on Cosmetics Safety, has an absolutely outstanding team behind it. My former PI on the NICEDOM support contract, Dave Allen, has recently left after 20 years with NICEDOM and ICFM and joined ICCS. So just, love that strengthening of those ties and collaborations there. HESI is another organization, the Health and Environmental Safety Institute, that brings together companies and academic researchers that are working more in the sort of environmental chemical space. The IQ Consortium, that's really advancing a lot of different aspects, including microphysiological systems in the drug development space. So having public-private partnerships that bring together consortia and government and academia is, I think, the most powerful approach to implementing change, and then working through existing structures like the OECD that are really designed to promote international harmonization and have legal statutes that really enforce that.
Thanks a lot. And Surat, we haven't heard you yet.
Yeah. So regarding if legislation is the biggest barrier, so like I mentioned, like India, the amendment was passed last year, but I would say legislation is definitely a significant step, but it's definitely not sufficient to ensure implementation, because although that amendment has been passed, I'm not sure if somebody has actually submitted the data to the regulators using a non-animal methods in India yet. So, but it does kind of open several doors, because earlier when we used to talk with the industry stakeholders, they often used to comment, what is the point of developing? Because even if we develop, it won't be accepted by the regulators. So that used to be the standard, statement given by them. But now, they don't have that kind of hurdle to fall back upon. So now we can also go to, for example, even the government stakeholders. And recently in India, the Department of Biotechnology also released a call last year, call for proposals, which included the development of human-based technologies for drug safety assessment, efficacy and safety assessment. So it also kind of, I wouldn't say it is sufficient, but it does help to accelerate the development of methodologies, because then now we can go to the governments and say that this provision is there, but then we need to do additional work to implement it. So they need to release a dedicated funding, call for proposals to develop these technologies. For example, in India, we can go to the technology developers and ask them, what their status is. And so I think it does help to accelerate, but it's, I wouldn't say it's the biggest hurdle. I would say it's the first step, which helps to kind of, opens the way for doing the work. So yeah, I think once that is open, you can help to kind of, work with different stakeholders and get the other barriers. You can work with that. So that's definitely there. Yeah.
Leela, can I ask one more question? Because the OECD should be or is a very important organ for the harmonization globally. So when the OECD has accepted guidelines, what I understand, then everybody knows they are good quality guidelines. So how fast does it move into national and regional legislation then? Do we have any examples of that, or how does that work? I don't know. I'm just curious.
So, I can start, because there's a difference between OECD member countries and non-member countries. Okay. So for OECD member countries, they are legally required to accept data that is generated under OECD test guidelines. Okay. It's called the Mutual Acceptance of Data Clause, which is part of the reason that it's a often challenging process to get a test method codified in an OECD test guideline, because you have very high standards, rightly so, of validation and reproducibility and rigor, and it requires consensus. So there's no such thing as majority voting in the OECD test guidelines program. It has to be 100% consensus, because you are requiring the countries then to, be legally bound to accepting that data. But I always like to say that MAD stands for mutual acceptance of data, not mutual acceptance of decision. And so that's somewhere where there's still some divergence, right? So you can have a test guideline that provides data that can be used to support a regulatory decision, but a risk assessor in one country may use it differently and may then require additional data before reaching a decision. And so that's something where I think, for example, the WPHA is a really good forum for communication among risk assessors and regulators for their processes and how they are integrating different lines of evidence and how they come to decisions. Now, for non-OECD member countries, there is no legal obligation to accept that data, but increasingly countries like China, for example, that are OECD observers, are becoming more and more open to following OECD test guidelines and recognizing their validity in accepting information. There's still a little bit of a challenge of needing those tests to be run in those specific regions. So not necessarily accepting data from an OECD test guideline that was run elsewhere. Like, they still want those tests to be rerun, but those are also driven by economic motivations. Yeah. So there's only so much we can control from a scientific and policy perspective, yeah. Right? But I do think that in regions like India that are not OECD member countries, but really have a tremendous amount of power to influence, globally what's going on in the chemical testing space because of the size of the economy and the amount of commerce and the amount of production that happens in those regions. There has to be really, really dedicated education and training programs to sort of socialize the message around not only the validity of NAMs, but the superiority of NAMs in particular endpoints and areas where, going back to what I said at the beginning, we actually have a responsibility to the public to be using the best science to protect human health and the environment. And in a number of examples, particularly topical toxicity examples, we have tremendous evidence that shows that the animal studies are insufficient, and we should be using human biology-based methods to ensure better public health protection.
Thanks for the great explanation about the OECD. So, Leila, back to you, or do we go to the questions of the public, perhaps?
Maybe, Surat, you wanted to say something about that too.
Just to kind of add on to that point, so like Nicole mentioned, India is not an OECD member, but so it's not automatically obligated to follow the OECD guidelines, but in several cases the Indian guidelines do mention so they have to categorically mention in the regulations or guidelines that they accept OECD particular guidelines. For example, for agrochemicals, the revision in the 2017 revision to the guidelines, there is one sentence that mentions that any OECD approved alternatives data may be submitted to the the registration committee. So it has to be categorically mentioned in the regulation that it accepts OECD test guidelines. So yeah, so that is just wanted to mention that. So for agrochemicals, they do mention that. Again, it's not like automatically accepted, but if someone can use those methods and submit the data, and it will be considered by the the registration committee. So I just wanted to add that.
Thank you so much, Surat. Yeah, that's very useful to know as well. I wonder, just being careful of time, if we, I will try to merge the two last questions, if that's fine with you, Meryl. So basically, it's a bit more philosophical, but we were wondering if we could learn from history. So is there any existing path that we can follow or should we just reinvent from scratch this path for NAMs? And I was wondering if you have in mind a really good success story that could be a nice example to follow, and where in your opinion, the reason for this success. And to finish, if you have any hopes for the future and expectation that you would like to share regarding the harmonization, please do feel free to share it with us.
Yes, definitely. It was already mentioned from Nicole and Surat. It's, for example, the monocytes. There are clear example, and I think this is exactly, people really have a lot of respect from OECD, and it's a lot of effort. And to get one, it's maybe also for some people still, including me, a little bit of black box how much you really invest, because it's not that you do it on your own. You need parallel studies, you need feedback. It's not happening just, okay, these months we do this, like an audit, which we just know, okay, we want to have an ISO certification, so we do it. It takes a little bit more, and maybe more parameters which you cannot totally control as a single unit, which is also should not be like this, because it should be general applicable. But I'm very sure if we go, I just say maybe it's important to also have little steps between, and there are little programs like iSTAND and the RFI, which I mentioned. But I'm very sure having an OECD appoint for your application for a certain case, for a certain question, is definitely what would be, which makes it then implemented. And we have beautiful cases about it from skin and from inflammation. There are cases which it was absolute successful, and people just use this anymore. Don't use any animal anymore. So I think there, and having this funded, of course, because of parallel studies are very expensive, and nobody feels responsible, or as a developer, we don't have so much fund for this. Having it funded would maybe drive it most quickest that way. Happy to give it on.
So I don't want to be repetitive of what I've already said, but just to sort of re-emphasize the point that as far as like topical toxicity endpoints are concerned, for skin sensitization, skin irritation and corrosion, and eye irritation and corrosion, the science is there. It's superior to the animal tests. It's based on human biology. It's more reliable, more rapid, more efficient, and more informative. And so we have a responsibility to implement those worldwide. We have OECD test guidelines for all of those methods. In particular, skin sensitization, we have a defined approach guideline. That's the first kind of full replacement for an animal test for hazard and potency categorization. And we have work ongoing to validate a defined approach for deriving a point of departure for quantitative risk assessment. And so having those in internationally harmonized test guidelines, the next step is to sort of demand implementation of those, and that can come from various sectors in various forms. So companies that are submitting data to regulatory authorities, they need to communicate within their companies and tell their lawyers that actually this is the better approach. This is the more protective approach. This is the preferred approach. And so this is what we should go with. Even if not every single sector in the world is going to accept it, we have a responsibility to the public to use the best science. And so communication strategies coming from multiple different arms, I think will really facilitate that. That would be my sort of short-term hope and expectation around harmonization of NAMs, is true widespread implementation of those NAMs that have been demonstrated to be superior for acute topical toxicity endpoints. Looking ahead to get us to a place where we at some point could eventually aim towards full replacement, we absolutely have to have increased investment and funding and resources into developing toolboxes that will help us get at more chronic, long-term toxicity endpoints like carcinogenicity, like developmental toxicity, like reproductive function. And that's going to take a lot of work and a lot of effort. And I think there's multiple ways that can be approached, both from a predictive standpoint, so to be able to get at sort of mechanistic methods that give us insights into prediction, but also from a protective standpoint. And there was a question in the chat from Zhongmin Choi on sort of NAMs that are practically applicable for general risk assessment. We have a new project under the OECD WPHA on developing a scientific confidence framework for approaches to risk assessment for systemic toxicity. And so that at a high level looks like, okay, what in vitro assays can we use that provide comprehensive enough coverage of biology, so high throughput transcriptomics, other, high secondary pharmacology screens, large scale high throughput screening, cell stress assays, etc. Apply in vitro to in vivo extrapolation, so use PBPK models to derive, equivalent administered doses or predicted exposures that would result in internal concentrations where biology is being perturbed in a potentially adverse way, and then take those derived doses and compare those with human exposures to create an exposure-driven risk assessment and derive margins of exposure that are protective, that maybe don't give you exact insight into exactly how this chemical or what specific target organ or what type of toxicity may arrive, but you have high confidence that if you're using this in vitro toolbox to derive a safe exposure, that is truly safe and that you are covering sort of all potential pathways of toxicity. And if we think about it realistically, that's all an animal test is doing anyway. If you look at the 90-day test, or any other chronic or subchronic type of in vivo toxicology test, yes, you look at something like 42 different tissues for, pathology, but you don't get at the mechanism of why a compound might be causing those particular effects. It's sort of the ultimate black box. And because it's a whole organism, it's assumed to be highly relevant and protective of the human scenario. But we have lots of insight from the drug development world and the pharmaceutical space that is not always the case. So if we can derive human biology-based toolboxes that are sort of equally comprehensive and equally protective, then I think that's a really promising approach for sort of general risk assessment.
I think Nicole and Nina have covered most of the great points. So just to I think cosmetics was a great example where several countries have now managed to get the ban on it, but at the same time it is based on a topical endpoint. So and it's a much simpler case study compared to some of the complex organs or systemic toxicity that we are looking in the case of drugs. So it's definitely a different case study, but it does provide a good example of how legislation can act a propeller for innovation, because the ban was enacted when in several countries we didn't have the alternatives. The ban actually accelerated the development of alternatives in, for example, in countries in India, to develop that infrastructure, develop the training, and get the countries equipped to develop those methods, like indigenously develop, get the training and have those methods developed in those countries. So it is a great example of how that ban could accelerate the development of methods. So so that's a great example. So other than that, I think if because the main which, as everyone has mentioned, we could probably focus on few priority contexts of use globally, so that different countries, because right now there are so many technologies, and each technology, so the approval and harmonization and the guidelines are so context specific. So globally we could agree upon 4 or 5 contexts of use, and if develop the guidelines and the acceptance criteria, so that it kind of gets it easier, to understand what is availability and how do we have like defined set of guidelines to approve that methodology. So I think, like for example, liver toxicity or cardiotoxicity, those are organ specific toxicity. So at least like EU has, for example, prioritized some of these endpoints. So at least we could start off by some of these prioritized endpoints, develop, have like some kind of harmonization guidelines for these some of the prioritized endpoints, which could then build confidence for these methods and again further propel the innovation in other sectors as well. So yeah.
Thank you so much. I think we reached the point where we will be willing to take time for questions for participants. Emmeline, if you want to, I think we have just one yet. So please do ask question if you want, and we'll try to answer. If we don't have time, we will probably see if we can answer later. But maybe, Emmeline, you can read the first question from Kevin, if you want.
Yes, exactly. So from Kevin Gillois, if we consider organ-on-chip systems, for example, how could we imagine an harmonization? If we consider only commercially available systems, I imagine that each company would defend their own system as the most adapted for a specific biological question. But if we consider the same cell model and, for example, a cell culture realized on an alveolic system or emulate one, endpoints results could be quite different. Maybe it is something that has been touched upon in the question too a bit, but if you want to go further.
Yeah, I think it's fair to comment on this, because I think it's really something, and it's good and bad. The organ-on-chip ecosystem and also organoids and MPS, it raised pretty dynamic, and it is really an ecosystem. So, and it's great. Many cool developers, academic research. So many stuff comes up. But of course, even it drives innovation and being better when you have competition. So competition is good. It also is then more difficult to harmonize. So let's assume that the history of organs on chip would have been different, that there would be one big research institute would do all organs on chip, and now they would go, for example, to the OECD committee and would say, now we have to harmonize it. The harmonization step would have been easier because there is just one system in place and everybody can now use it. And I think this is the burden and the benefit that we have. It's definitely not meant, and none of the organs on chip companies would think about this. The tool might be different, but the common unit we have to find on the endpoint. So let's assume emulate system for one endpoint which is shown as being clinically relevant and important has the better outcome. It shows this effect more like in human. Then the emulate system is the better for this for this translation. But on the other stuff, maybe tissue, maybe alveolix is the better. So we have to harmonize on the endpoint and on the readout and the clinical relevant endpoint, and might not can harmonize on the tools that you use. And I think this is also, and I'm happy that Surat and Nicole can also correct me, but from everything that I know about OECD, they try, of course, to tell you which flask you should take, and then you try to see which kind of flask medium was taken. But technically, it's still not nailed everything in stone how you get to this perfect readout. So therefore, I think it's really the challenge and the benefit that we have. And yeah, it's a very relevant question. I think it's very also important that people don't think that we do monopoles or so, that anybody will go for a monopole. It's something which is for the public. So always the best case should win because it's for public health.
Yeah, I would like to add to that, I mean, that's essentially the philosophy behind key event-based test guidelines at the OECD. So there are a number of different analogous or orthogonal methods that are measuring the same biology. And so the expectation and one of the reasons that there exist things like performance standards, reference chemicals, is that if you are measuring the same biology, but you're using different technology or different platforms, you should still arrive at the same answer most of the time, right? And so I think the same is true for MPS, even though those are significantly more complicated systems than some of the, protein reactivity assays or simplistic assays that fall under some of the key event-based test guidelines. The same approach should still be applied, but that's where I think there is a lot of work that's needed to define reference chemicals. So to do, and this is, we didn't get to this question earlier, but I do want to bring in sort of the utility of artificial intelligence here in a variety of different applications. And the one I'm thinking of right now is the ability to synthesize massive amounts of information from the scientific literature and aid in things like systematic evidence mapping or systematic reviews that will then allow us to derive sort of reliable reference chemical lists. So here's chemicals where the weight of evidence in the literature from in vitro, in vivo, human epi studies, et cetera, really points to this chemical at this dose is, expected to cause this type of toxicity in humans based on human biology. And then once we have those reference chemical lists for particular target organs like the liver, like the kidney, like the lung, then we can start to do validation studies that look at consistency of predictivity and response across different commercial systems that are intended to measure the same biology. And NCATS here in the US, the National Center for Advancing Translational Sciences, that has, really been the front runner, I think, at least in North America, in terms of developing and funding tissue chip programs. They have a new funding program called TRACE, which is the Translational Centers for MPS, that's really designed to do exactly that, to develop or validate tissue chips that will meet criteria for FDA regulatory qualifications as drug development tools. So I'm really excited to see what comes out of that program.
Just to add to that, so like any device like Organ on Chip, it will have like basically I think several workshops are now divided it into two broad areas, like the engineering aspect and the biological component. So the engineering aspect, which are basically the the internal all the engineering aspects which are non-biological in nature. So that aspect will not depend upon the context, ideally not depend on the context of use or what the endpoint it's being tested for. So those guidelines and standards should ideally be consistent across different devices. And the biological component, which is supposed to assess the particular endpoint, that could, of course, there are different parameters which Nicole mentioned using reference compounds and the key events which could be measured using for the biological components per se. So,
thank you so much, and thank you for touching about the AI. It's a bit frustrating because we would like, at least I would like to ask a lot of other questions, but I think we covered quite a lot of things, so that's really good, and it will definitely deserve a bit more discussion, but hopefully we will have another time later. And I'm conscious of time, so I think we can finish by thanking all of you, and I'll give the floor to Emeline to conclude, but thank you so much for this great discussion.
Thank you.
Yeah, thank you. I will just thank you. Yeah, thank you again, all of you, for making this session possible, our panelists, for this very rich and stimulating discussion, our moderators that also bring value to it, our partners, PARC, the EPSI-FRIA and the ATC, and of course the participants. Do not hesitate to send us any feedback you may have. It will be very much appreciated. A replay will be available soon, and we will be pleased to keep you informed on the following and the next session. So thank you very much. And we wish you a great afternoon. So thank you very much.
Thank you.
Thank you. Speak soon. Bye.
Thank you. Have a good afternoon.
Thank you.
Bye. Thank you.
Bye-bye.